The DNA replication begins with the separation of the double helix structure of the DNA molecule. The enzyme helicase breaks the hydrogen bonds between the nitrogenous bases and creates a Y shape called the replication fork. Single-strand binding proteins bind to these unpaired strands to keep them from repairing, and the topoisomerase relieves strain on the DNA that is overwound ahead of the replication fork by breaking and rejoining the strands. This results in two parent DNA strands that will serve as templates for new complementary strands. Next, primase makes RNA primers on both of the template strands, and the primers serve as starting points for the DNA polymerase to begin their work. DNA polymerase adds DNA nucleotides in the 5 to 3 direction to build the new complementary strands. Since DNA is anti-parallel, the two strands must be synthesized differently. The leading strand can be synthesized continuously because DNA polymerase works in the same direction as helicase towards the fork. However, the lagging strand is synthesized in segments called the Okazaki fragments because as DNA polymerase replaces RNA primers with DNA and adds nucleotides away from the fork, helicase unzips more DNA in the opposite direction. Finally, DNA ligase joins together the Okazaki fragment on the lagging strand, and DNA replication results in two identical copies of the original DNA. The two daughter molecules each have one original parent strand and one new complementary strand, which follows the semi-conservative model.